Now let's talk about the life cycle of an RBC. Now we have up till this point studied that how erythropoiesis happens, how RBCs are formed, how they are regulated, what is necessary for their regulation, what is necessary for their formation. But what happens once a reticulocyte matures into a erythrocyte? After the formation of erythrocyte or a mature RBC, what is the life cycle of it? We will study that point in this bite. So let's get on with it. The life cycle of the RBC. Now, we know that there are certain points in our body which are designated as the hemopoietic sites. These are the locations of the hemopoietic bone marrow. Now, usually the sites of the hemopoietic bone marrow is the distal ends of the long bones and the bone marrow of the membranous bones, such as the sternum, rib, and the body of the vertebra. So, at these locations, what happens is that there is a stem cell, and that stem cell converts into an erythroblast. That erythroblast again is of the erythroid lineage, that again converts into the reticulocyte. Now we have studied all of these three phases in much more detail in another bite. Now once this reticulocyte has been produced, what happens is that this reticulocyte comes into the circulation of the blood. Now it comes into the circulation of the blood via process known as the diabetesis, because reticulocyte has a shape like a bag, and because it is shaped like a bag and about half of its space is empty, it can squeeze through even the tiniest hole of a capillary. So from the bone marrow it squeezes out of them and into the capillaries of the systemic blood circulation. As you can see easily here, the reticulocytes are released from the bone marrow into the bloodstream, where then they further mature into the RBCs. Now, once the RBCs have matured into a proper erythrocyte, they circulate in the bloodstream for a total life cycle of 120 days. After the 120 days of their life cycle has expired, they are destroyed. So, after their 120 days of life cycle or 3 months, what happens is that all RBCs after that much duration have lost their shape, they have shrunken, they have lost most of their ability to carry the oxygen. And at this point, all the old and damaged erythrocytes are phagocytized by the macrophages. Now, macrophages are found both in the bone marrow, liver, and ultimately in the spleen. Spleen is the final destination of the destruction of all RBCs. And these macrophages, what happens is that they try to engulf the damaged, shrunken RBC, and once they are engulfed, they are broken down. So, once they are broken down via certain organelle known as the lysosome. Lysosome contains certain enzymes or lysosomal enzymes which are mostly proteolytic in nature, and because of this, the hemoglobin is released from the damaged and old RBCs, and then the hemoglobin is further broken down into its heme component and the globin component. So once they are separated, they go their different way. The heme component, which is found 4 times in one molecule of hemoglobin, and the globin part, both of these are broken down into their smaller unit. The globin part is broken down into the building blocks of the proteins, that is the amino acid, while the heme part is then further cleaved down into the porphyrin rings. Now once both of these components have separated, they go their own way. So when they go their own way, the heme part is then further divided into the heme group and the iron ions. So porphyrin rings separate from the heme part, that is the heme groups, and because the porphyrins have released the iron is released into the bloodstream. So we can say that hemoglobin is then divided into two parts, the proteinaceous part, that is the globin, and the metallo part, that is the heme. Heme has further two parts. One, the porphyrin rings, which are four, and the ferric ion that is found inside them. And because of the lysosomal enzyme, all of these components are broken down. Now, globin, along with their amino acids and cell components, is then further changes into the much more smaller building blocks. Now, the iron ions then bind with certain transporter proteins which are found in the blood, such as transferrin. And because of the transferrin, these released iron ions then transfer from one part to the another. Usually, the role of transferrin is to take the free iron and store it at certain sites in our body, while the heme group is then further converted by the release of its porphyrin rings into the biliverdin, and that biliverdin is then converted into the bilirubin. Now, once biliverdin has formed, what happens is that it converts into the bilirubin. And bilirubin, as we all know, is the excretory product majorly associated with the degradation of the heme. And it is the bilirubin which gives the yellowish appearance to both urine, and it is the biliverdin that gives the greenish appearance to the bile. So all of this degradation of heme usually takes place in the liver. Liver is the site of the degradation of the heme component of the hemoglobin, where iron is taken by the transferrin protein and then stored, while the heme part is then converted into biliverdin, which is then excreted into the bile, and it gives the green color to the bile, and then it is converted into the bilirubin. Now, when bilirubin is then further converted into either stercobilin, urobilinogen. There are a variety of other pigments that can be formed after this. But bilirubin is usually considered the final end product of the heme degradation, because then it is released both in the urine through the filtration of the blood and in the fecal matter through the excretion of the bile. And the iron, again, via the transferrin protein, will either get stored inside the tissue of the liver through the ferritin, or it can go again through the cycle of the hemopoietic sites. Now, if it goes to the site of the storage, it will be stored. If biliverdin goes to the ways of bilirubin, it will be excreted, or biliverdin can, instead of being excreted, can go to the location of the hemopoietic areas. From there, it is then further utilized to form the heme group. So the heme group can be further regenerated from the biliverdin. The iron, if it goes to the bone marrow sites, gets locked in the heme site, and the globin, amino acids, all of these, when they enter the bone marrow, they form the, again, either they are reused or new globin chains are formed. And because of this, the globin part is regenerated. So as you can easily appreciate, a cycle forms. The cycle starts from the hematopoietic sites to the all the way to the diabetes to the maturation, then the phagocytosis of the old and damaged erythrocytes, then the split of the heme and the globin parts, then the ultimate fate of both the heme parts, the iron parts, and there again the further usage. Bilirubin might be get excreted or it might be get used again. So this cycle continues until we have enough RBCs, and it can go to the storage route if we do not. Thank you so much for watching sqadia.com. You can find lectures on topic ranging from biochemistry to physiology, from anatomy to rheumatology to courses in medicine, surgery, radiology. You can find more than 1400 lectures on sqadia.com. You can even start your free 2-day trial on this site. You can even download the app both on the iOS and the Android, and you can start your free trial. So once again, thank you so much for watching sqadia.com.